about public water exposures from exposure assessment to epidemiologic applications. And I will definitely finish with some time for us to just chat and have some questions at the end. So yes, like Amy said, the long-term goal of my research team is to reduce inequities and exposures and adverse outcomes. It's a very broad and very ambitious goal, but that's what we're aiming for. And we do that by kind of basing our work here. So we do a lot of work on drinking water, we do a lot of work on metals and biospecimens and in other media, and we focus our work broadly around these four areas. So we do classic kind of epi stuff to inform risk assessment. We do policy and regulation evaluations. We do a lot of work on exposure assessment, and we do a lot of tribal community directed work. We have a training program, we do research, and we do intervention planning. So I'll touch on a couple of these things, right? Some of these are very translation epi relevant, especially the work on policy and the work on intervention planning with tribes. So I'll give you kind of maybe a highlight reel of a couple buckets, a couple examples of how our work fits into these buckets. So if you're in the US, this is just a little bit of background information about why should we care about, regulated public drinking water. If you're in the US and you have water that's piped into your home, you're getting it from either a US private well or from a regulated public water system. And these regulated public water systems serve most US residents. Over 90% of US residents get at least some water from a public water system, and they're federally regulated by EPA under the Safe Drinking Water mandate. Depending on how you count them, there's now over 90 different federally regulated contaminants in our public water systems. And those systems are really required to conduct routine compliance monitoring to ensure that they're compliant with those EPA standards. And so when I was training in the field, which was not that long ago, the narrative in our field was really that we focused a lot on private wells and private well users, but we pretty much ignored public drinking water as a relevant exposure source, except maybe for lead. There was still a lot of work going on lead. But the narrative in the field was really that if you were connected to a public water system, then you were really protected from exposure to contaminants at levels that would be concerning for health, because your water system was being, monitored and regulated by EPA. Of course, we know that that's not true. So every couple of years, there's a major water crisis that is impacting a public water system somewhere in the U.S. And there's a, headline that hits our national news media, and we're all reminded that this is not, in fact, the case. So on the left here, we have an example from the Jackson water crisis. We have a new RO1 that's evaluating water contaminant exposures in the Jackson Heart Study in response to that crisis. Here on the right, just an example of Latino and Hispanic communities in the Southwest being heavily impacted by arsenic and other geogenic contaminant exposures as well. So this, remains a very salient and important environmental exposure and issue, and tends to impact communities that are less well-resourced, that are in rural areas, that are, serve minoritized or racialized communities. So certainly more work to do. And if you've never seen what the national primary drinking water regulations look like that EPA sets, here's just a screenshot to give you a sense of just a couple different contaminants, alphabetically the first 5. And so it's important to note that this column here, which has MCLs or regulatory standards or treatment techniques, these values are what's allowable in drinking water. And there's another column over here with the public health goal for these contaminants. And these are quite different, right? What's allowable in drinking water, what the federal regulatory standards are, the treatment techniques are. EPA is mandated to consider cost and technical feasibility as well as public health benefit when they're setting those standards. And so there's a difference here between, for example, 10 micrograms per liter of arsenic is allowable in drinking water, but our public health goal, if we didn't have to consider cost and feasibility, would be 0. So there's quite a big margin here between what we believe to be quote unquote safe and what is allowable in our public water systems. So one of the areas that my team is really working on is to fill in this critical need in terms of doing kind of a geospatial exposure assessment for drinking water contaminants in big epi cohorts nationwide, right? So often I work with a lot of different cohorts. I'll talk to you a little bit about ECHO, but I work with a lot of different cohorts, and we have, people, and so usually we have at least one address, right? And we want to try to use a geospatial approach to assign them drinking water contaminant exposures. And in all of our work with all the cohorts that we've been working with, what we're finding is that having a single address, maybe with a zip code attached that was collected at baseline is very common, but it's very difficult to utilize. And it's not desirable to just have a zip code or a single address that hasn't been geocoded, right? From an exposure assessment perspective. That's a poor situation to find ourselves in. And what's very uncommon but highly desirable, right, would be to have a residential history file for a participant that's been linked or appended to census geography. So something like tracks over time, and we could do kind of easy column merges. The reality is that many cohorts, many large NIH-funded cohorts, simply haven't geocoded addresses at all, or have single addresses that haven't been appended to any census geography. So we ran into this real feasibility challenge quite early on a couple years ago when we started working in this space, and we've had to be quite flexible and adjust our exposure assessment techniques accordingly. And so I want to make a quick contrast here to what our field has done for air pollution, right, over the past several decades, which is, we've done an amazing job in the field of environmental health of, generating high quality, high resolution exposure estimates, quantifying uncertainty resulting from different, mechanisms, and decomposing that uncertainty across different components and using things like Bayesian ensemble models to deal with that uncertainty. And we don't currently have anything like this for drinking water. And the air pollution world has been quite successful in using that science with the really sophisticated exposure assessment techniques to push policy forward, right? So this is now, 2 years outdated, but that when EPA originally first proposed to change the NAAQS standard for PM 2.5, right, they cited a lot of the science that used this high quality exposure assessment work that has been ongoing for decades in our field. So this is just to say there's a lot of work to be done in this field on drinking water. So the premise of our lab's work is that we can directly inform public water regulations and policies by characterizing inequities and exposures and evaluating associations using a more geospatially assigned exposure assessment approach, which is much more feasible for a lot of these large cohorts. It reflects, temporal variability in exposures better, and it's just less challenging than doing, a high quality home tap water assessment for all participants in a given cohort. And it allows us a little bit of a time machine to go back and assign exposures that happened previously in the past. And so some other benefits of this approach of using public water system contaminant estimates themselves is, one, we don't regulate biospecimens, or, we don't regulate concentrations of lead in blood or arsenic in urine or lead in blood, really. And so when we are using in our science these exposure estimates that reflect how water systems are reporting in accordance with the Safe Drinking Water Act, there's a direct relevance there to the findings that I think is quite powerful, right? So a water system reported this particular monitoring record, and that's what we're using in our science. So that's very helpful. And then just from a feasibility perspective, again, these geospatial exposure assignments make it much easier to work in these large cohorts, very few of whom have collected a drinking water sample. And, as far as I know, none to my knowledge have collected multiple samples indexed over time that we can leverage to reflect the reality of time varying exposures across participants and communities. So this is just a handful of the cohorts that we've worked with, and I'll give you just a couple examples going forward. Okay, so bucket one of the type of work that we do, so exposure assessment work, right? One of the things we aim to do are to develop these nationwide estimates of regulated contaminant concentrations at different spatial and temporal resolutions, try to maintain flexibility in how we can do the exposure assignment approach so that we can work with as many different cohorts as possible. So they're readily assignable, we can work with, I'll show you, we can work with ZICTAs, we can work with tracks, we can work with shapefiles, we can work even at the county level. We worked at the county level. And we've done a little bit of work to try and validate this approach with biomarkers. And we're thinking next, our next steps, our next iteration, we're really focused on measurement error and trying to find ways to minimize measurement error, because we all know that's one of our biggest challenges and potential sources of bias in our environmental epi studies. And so that's very much on our mind for the next 5 or 6 years of work. So when we make these estimates in public water, we pull from a US EPA database that currently covers 2000 through 2019. And this particular database, again, includes these routine compliance monitoring records for almost all public water systems and most of the population that's served by public water systems. So you'll notice there are some states here that are grayed out, and we've, to deal with these states that didn't voluntarily submit data, we just web scraped those records for when they were available. So we've web scraped a lot of those records and been able to append what's available and extend also our available dates out past 2019. So again, for all regulated contaminants, millions and millions of records. So this is a big data challenge. And almost all community water systems are represented here, including tribal water systems. And so EPA requires water systems to conduct this routine compliance monitoring at different temporal scales or averaging periods, depending on the contaminant itself. So for example, for disinfection byproducts, something like trihalomethanes, systems are typically required to report at least once per quarter or once per season. So we have those estimates aggregated to quarters and seasons. And then for something like uranium, which is a radionuclide, EPA only requires at minimum 1 sample taken every 8 years, right? And there's differential missingness here by system size, by the source water type, by your last monitoring record. And so we select these averaging periods to minimize that differential bias by water system characteristics, so that when we make comparisons across systems, we've tried to minimize differential missingness that way. And to spatially aggregate, we've intersected water boundaries with different census geographies. We've incorporated, for ZICTAs especially, US Postal Service and census data, and we weight by the underlying census population so that when we aggregate to a tract or we have blocks, we're weighing the contribution of each water system to the tract by the number of people that are served by that water system. So we have these at the water system level. There is a national shapefile of estimated water system boundaries across the US. Some of these are very high quality, some of these are rather low quality, but it does exist. Various census geographies, ZICTAs, and counties. So just to give you an example of what this looks like when we move, here's an example in Texas. It's arsenic from a recent time period. And as you move from public water system boundaries, Texas has a rather high quality shapefile here. So this is one of our more high quality states in terms of the shapefile boundaries. When you move from public water system to a census tract to a ZICTA, right, you can see the overall spatial patterning here changes. And I want to make a note about a big gap that's currently in our field that we're also trying to address, which is that we don't currently have any concrete evidence to support which of these levels, or county, right? I didn't put county on here. I could have put county on here. But we don't have any current evidence to support which of these spatial levels is actually the most appropriate for estimating public drinking water total exposure for a given participant and for a given address. So if you think about where you might get your drinking water from, your primary source is probably your home, but then you have secondary sources, places where you work, places where you go to school, places where you worship, places where you pray. And we don't actually have the type of validation studies done yet to support selecting any of these particular spatial resolutions as more closely aligned with a gold standard measure. We haven't really even defined as a field what a gold standard measurement would be for total public water exposure from all primary and secondary sources. So that just remains an open question in the field that I think is really, really interesting. And I'll note that, these exposure patterns across the US broadly are very different by contaminant and by time period. So here I'm just showing you arsenic and fluoride. which are actually like relatively similar as terms as water contaminants go, because they're both inorganic, they're both geogenic. Fluoride, of course, is also added intentionally to water systems to prevent dental caries, but on the whole, you'll see even these two geogenic contaminants, right? There's spatial differences here as well in terms of those patterns. Arsenic matches pretty closely to underlying source water aquifers, and fluoride is a little bit more homogeneous because of the additional additions at the water facility. So there's no easy one answer to what do water contaminants look like spatially across the US. They're very different because each of these contaminants has a different source profile. We've done a lot of work looking at racial ethnic composition and other inequities in water contaminant exposures. And so this is at the ZICTA level, but it's really mirroring an earlier paper we did at the county level. So in this example, we're looking at associations between racial ethnic composition and the concentration of arsenic, again, in regulated public water systems. And so consistently, we've done this analysis now at the county level, at the ZICTA level, at the tract level, at the water system level, and we see the same associations, which is higher arsenic as the proportion of residents who are American Indian, Alaskan Native, or Hispanic Latino are also higher. And that's quite consistent. And in these models, we've also fixed our covariates here such that these, when you have higher or increased, when you have higher levels of higher proportions of residents who are American Indian or Hispanic Latino, that is specifically because the proportion of residents who are non-Hispanic white is lower. So that's telling us that these associations are really due in part to the absence of non-Hispanic white residents in the community. That's part of the story. So the next kind of set of studies that we've done in this space is to try to start to validate these geospatial estimates with biomarkers of exposure, which is one way we can try to, convince ourselves that these are in fact reasonable to use. And so this is a study that one of my prior PhD students did. She's now at NCI. But we used two different studies here, two different study populations. So one is the strong heart study down here, and one is MESA, the multi-ethnic study of atherosclerosis. And in this study, we looked at the contribution of our drinking water estimates to biomarkers that were measured in urine. And we did this for arsenic and we did it for uranium. Those are two water contaminants that we also typically measure in urinary biomarkers to get a good sense of estimate of total internal dose. And in our field previously, the narrative and the best available science we had at the time suggested that public water was just not really a meaningful source of arsenic exposure, especially if your water levels were below the regulatory standard. That was the narrative that we were really working with, and that was our best available science at the time. But we didn't have these actual estimates of geospatial estimates of what was measured in drinking water. And so there was a lot of, modeling and assumptions that were happening previously. So we looked to see, very basically, is there this association between what we're estimating in water and what we're measuring in urine, right, based on your address. And so here I'm going to show you the adjusted percent change. This is from a geometric mean ratio of urine arsenic for a twofold higher public water arsenic concentration. And here, this is a biomarker that is reflecting inorganic arsenic exposure specifically. And so in both cohorts, we see, if you have higher water arsenic, then you also have higher urine arsenic. But what was surprising to us was the percent of variability that we could explain in our urine arsenic biomarker with our zip code level estimates for water. And it was much higher than what we had previously estimated in the field. And this varied depending on whether we were using the urban cohort, which is the MESA cohort, or the very rural cohort, which is the strong heart study cohort, largely rural, more rural than MESA. And we think that reflects in part that we know some folks in strong heart are also using private wells, are reliant on private wells, and we don't expect that to be the case for MESA. We don't have great data on that in MESA, but we expect that most folks in MESA are on public water systems simply because where they were recruited from. So we did our best. We have some other things ongoing we're going to try to do to tighten up that validation piece of it, because I think it's really important and I think it's a big gap in our field. And we're constantly trying to find support to do those types of analyses, we'll say. So bucket two of the type of work we do, so we take these exposure estimates, right? We look at them inequities nationwide. We try to understand spatial patterns. And we've tried to convince ourselves that they're reasonable to use in epidemiologic studies. And now, next we start looking at epidemiologic associations, right? Linking these to relevant health outcomes in our large cohorts. And so I'm specifically really interested in looking at exposures, of course, below current regulatory levels. And I'm really interested in projects that can directly inform drinking water policy. And so, for example, EPA recently finalized a new risk assessment for inorganic arsenic. This was just finalized in January of this year. And in looking at that assessment, it's very clear to see where the gaps are, right? Where the data gaps are, where the epi gaps are. That EPA is kind of, asking us as a scientific community to work on filling in to help inform these types of assessments. And so, for example, the quality of the evidence, EPA ranks quality of the evidence as being, moderately confident or, confident. They use a lot of causal language, right? They're trying to make causal assessments about things. And so my team, we read that type of documentation very carefully to try to understand which studies we can do to support filling in those gaps. And so one of those gaps that I'm going to show you for arsenic was around diabetes, type 2 diabetes, and another gap was about reproductive and birth outcomes. So I'll show you just an example, two different papers we've done to address those two specific needs. And most of the limitations that we, have that were identified for example, for arsenic were essentially, we didn't have a lot of studies conducted in the United States at, typical exposure levels that would be common in the US, and very, very few studies that measured arsenic, for example, in drinking water. And so for each of these outcomes, there's different gaps like that that we're trying to fill in. And very few that focus on public water as well. A lot of these studies have been conducted with folks who use private wells, which is very important, but their exposure levels tend to be higher. Again, I care about public water in part because we already have a regulatory infrastructure in place for public water, and so it's a little easier for us to think about modifying those exposures through regulatory action. Okay, so the next analysis we did in those same two cohorts, right, in MESA and in Strong Heart Study, was to just look at public water arsenic and incident diabetes. So diabetes and other metabolic outcomes, it has been traditionally, again, when I was training, which was not that long ago, it was somewhat of a contentious kind of space, right? Whether arsenic was associated with diabetes, whether it wasn't associated with diabetes, was it confounded by seafood intake? This was a really challenging question for us to think about, those of us in the arsenic community, because a lot of our studies were relying on arsenic as it was measured in biospecimens, as it was measured in water. And there are, reasonable, significant concerns that when you measure arsenic in urine, what you might actually be reflecting, you might have a reverse causality issue. You might actually be reflecting kidney function and excretion of arsenic out in urine. And it can be quite challenging to control or adjust for seafood intake and other non-toxic arsenicals that are picked up in our biomarkers that don't necessarily reflect inorganic arsenic or the most toxic forms of inorganic arsenic. So those are some of the challenges that we have with using a biomarker, some biomarkers, especially for an outcome like incident diabetes. And so having this public water arsenic data available that we could leverage for the exposure assessment here is exciting, right? Because we can kind of account for those challenges. There are other challenges that using water instead of urine introduces. But at the very least, we can deal with the reverse causality concern. We can deal with the confounding by seafood concern, and focus on what's coming from water. So of course, this isn't total inorganic arsenic exposure that we're modeling here, right? This is just water arsenic. So we're missing any contribution that might be coming from rice intake or other dietary sources or air pollution, right? But I think this is very helpful for the reasons that I stated, to get around using biomarkers. And so here, again, MESA is our urban cohort. So we have participants in urban and semi-urban areas across the US at multi-site cohort. Strong Heart Study is a rural cohort of all American Indian adults from 3 different study sites. And here, this is just, I'm going to show you the hazard ratio of incident diabetes for a doubling of arsenic levels in community water systems. And another challenge that we needed to address here, it's not really a challenge, but a fun kind of complication, is potential modification by BMI. So there was also evidence from the literature here that inorganic arsenic, the association with type 2 diabetes, might be modified by BMI. So we knew a priori ahead of time that we would look at potential modification by BMI, and we thought that would be really relevant. And so overall in both cohorts, we see this positive association: higher water arsenic, higher hazard ratio of incident diabetes. We certainly saw effect measure modification by BMI, so larger effect estimates among the leanest participants, or participants with the lowest BMIs. And we used a pooling approach to combine these 2 cohorts. And so for a log doubling in water arsenic, we get a hazard ratio of 1.10, which is also very similar, actually, with the magnitude of the effect estimates that we see when we use urinary biomarkers of inorganic arsenic. So that was, I was happy to see that. That reassured me, at least, that we weren't, getting wildly different magnitude effects from these 2 approaches. Okay, the other project that I have been working on with Amy, which is very exciting, Amy, who's obviously working at the DAC for ECHO, the Data Analysis Core for ECHO, and this is the first paper that was just accepted last week, I think, so it's still in press. But another big data gap that you can see in EPA's risk assessment for inorganic arsenic is on reproductive and birth outcomes. So I mentioned that earlier. And one of the reasons EPA judged that evidence to be, they're moderately confident that there's an association, right, like a causal association between inorganic arsenic and adverse birth outcomes, but they specified that there were major data gaps, and those data gaps included not having US populations represented at exposure levels that would be relevant today. And very few studies, again, if any, that leveraged public drinking water estimates. So we could take these estimates and they would be directly relevant for policy and regulatory action. So we brought in our public drinking water arsenic estimates here into ECHO cohort. So this is almost 14,000 infants from 35 different cohort sites. You can see that most participants came from a handful of states here, right? I want to call your attention here to the fact that the regulatory standard for arsenic is 10 micrograms per liter. And in the ECHO cohort, we had very, very few participants that exceeded the regulatory standard. Maybe none, maybe 1, maybe 2, very, very few. And so we really had most of our participants well below the regulatory standard of 10. Here you can see the darkest red areas are where the water arsenic concentrations are above 5, which is half of our federal standard, but it is the current standard for the states of New Jersey and New Hampshire and Denmark. And the Netherlands actually sets a standard of 1 microgram per liter. So these are low concentrations, but still in a regulatory relevant space where we have individual states and other nations that have established more health protective standards than what we have nationwide. And so we looked at a variety of different outcomes. Right, here's, I'm going to show you this spline for low birth weight, and I'll explain the spline to you. But this is here the relative risk of low birth weight for these 14,000 infants. And you can see, again, we had a lot of participants with very, very low water arsenic. We set our reference here to what's essentially the limit of detection divided by the square root of 2. The spline is not quite linear, right? The shape of this is not quite linear. It's not clear whether that's real or not, or whether there's some confounding happening here in the middle of the exposure distribution. But an important thing to note here is this relationship looks like there's an increased risk of low birth weight as your water arsenic gets higher. But we still had very few cases. We had very few participants with water arsenic above 5 and very, very few cases. So it's a limited kind of analysis here. I would say limited by precision, by just a low number of cases. And so if you instead just look at the continuous versions of this outcome, right, instead of looking at low birth weight, but birth weight itself amongst all participants, and then birth weight for gestational age Z-score, the shape is actually, the same, right? The shape of the dose responses or exposure response is the same. And so that is very reassuring as well, right? That we're not necessarily limited here just by cases for this particular outcome. The other thing we really focused on was looking at, again, potential effect measure modification, differences by subgroups, which is always important for my team to look at. And so if in a linear model where we're looking at the relative risk for a 1 microgram per liter higher water arsenic, which again is a very small, jump over here on the exposure distribution area, overall our risk ratio is 1.03. And we have, higher effect estimates for infants that were born to white moms or Hispanic Latino moms. And these types of differences we saw across the other outcomes that we looked at as well. So preterm birth and reduced gestational age. So exciting paper, and also really set the stage for a lot of the other work that we're doing now. So thank you to Amy, and I think I saw Mohammed's name in the Zoom. Mohammed worked really hard on this at the DAC, so thank you both a lot, because I think, what we've done with linking these water estimates in with ECHO is proven to be really generative and really fruitful. And we have something like 10 other APs that are, in progress right now, working on all different types of exposures. So we have APs on trihalomethanes and fluoride and other metals in water, and the outcomes range from NIH Toolbox cognitive scores to infant growth trajectories to Justine's leading a paper on the microbiome. So just exciting to be able to work with the ECHO, with DAC and with Amy to make this kind of stuff happen. Okay, so the third bucket of work that we do in my team, which it's a brand new project. We're still in year 1, so I'm very excited to just tell someone about it, even though I have no results to really show for it now. But I'll just tell you how we're thinking about this. So community-driven intervention planning. So we do a lot of community-directed research. We do a lot of community-directed training. I have an R25 research program that does some work on metals and drinking water as well with high school students from communities in the Northern Plains. And this new project we have is very, very exciting. So it's a brand new project. We're supporting 12 individual tribal communities in developing targeted intervention plans to reduce water exposures. And I'll tell you a little bit about that, and maybe I'll catch up with you in like 3 to 4 years and tell you about all that we've accomplished. So this is a new center we have that's based out of the University of Oklahoma. It's called the ICON Center, Improving Cancer Outcomes in Native American Communities. And I lead this one of our projects, which is called Water for Health, Strengthening Tribal Action for Cancer Prevention. And so we've done a lot of this water work in the Strong Heart study, which I mentioned earlier. And the Strong Heart study is the longest and largest epidemiologic study of American Indian adults, and folks are followed primarily for cardiovascular disease. And I've been working with the Strong Heart since I first came to Hopkins in 2014, so over a decade ago. As a master's student, I've been working with the Strong Heart study. And one of the things that we've identified over time is that many of our communities, especially those in South Dakota and Arizona, have high levels of arsenic and other geogenic contaminants in their drinking water, namely arsenic and uranium. And this is, you know, a real concern for communities, a real concern for tribes. And there's been a lot of work, including work directly out of Hopkins with Christine George and others, to develop interventions for individual communities to support them in reducing arsenic and uranium exposure. So this is a very community-directed center and a very community-directed project. So this particular project has 3 different aims, all focused around translating the water exposure assessment and epidemiology that we do into intervention planning for the 12 communities. So the first aim of this new grant, right, is we're developing individualized exposure assessments, not just in public water, but also in private water and in groundwater, which is relevant for a lot of our communities, and developing these community-specific dashboards for each of the 12 tribes. So doing high-quality exposure assessment, geospatial modeling, process-based modeling, combined with our public water estimates to generate, you know, dashboards for each community, so that folks can see, right, where we think, based on our best available evidence, where we think arsenic is really a problem, where we think uranium might be a problem. So providing more accessible information for, you know, leaders and other folks who are decision makers in communities. Then we have a very classic cancer epidemiology aim, right? So we'll be looking at our drinking water arsenic and uranium estimates with cancer incidence and mortality. And then the third aim is kind of leveraging all that information from aim 1 and aim 2 to help each of the 12 tribes develop a community-specific and community-driven intervention to reduce arsenic and uranium in drinking water. So there's 3 kind of phases to this. The first is that we systematically assess prior successful interventions, what's worked well, what hasn't worked well, and from a variety of different evidence streams. We're currently doing formative research, so key informant interviews with decision makers across all 12 tribes, which is a massive effort just to get approvals and IRBs in place and, you know, communities excited and on board. And then later on, in later years of the project, each of these 12 communities will be developing individualized intervention plans, in part via, you know, workshops and community forums. And so some potential possibilities that might come up, and, you know, we'll see what communities want to do, but some potential possibilities could be, you know, point of use filters in the home, extension of public water lines to folks who are currently on private wells or who are hauling water, tribal resolutions to lower water standards. Several of our tribes are very, very interested in that. And so many tribes can set drinking water standards that are more health protective than what EPA mandates. And so several of our communities have said they're very interested in that possibility. And finally, another possibility would be integrating home water assessments with healthcare, with point of care, for example, with IHS. So very excited about this project. It feels like the culmination of a lot of the work that we've done over the past 5 years into something that's translatable for, yeah, the communities that we work with. So with that, I have a lot of people to thank. This is broadly my team here of students and postdocs and staff. If you're looking for a great postdoc, I have several who are wonderful, and PhD students that are coming up. So please reach out if you're looking for a student or a postdoc or a staff member. I want to thank Amy and the DAC for all your collaboration on the ECHO work. It's been really fun and really rewarding. And funding from a lot of different institutes and all the cohorts that we work with. So thanks very much. Let's see.